Hello, everyone. Whether you are Jewish or not, Ashkenazi or not, the mystery regarding Mrs. Silberstein's ancestors could have engaged your mind. This video will show you that asking Mrs. Silberstein is not the optimal way to get an answer. You will find out that different answers have been around. Historians, priests, rabbis, linguists, reporters, individuals engaged in public discourse, and last but not least, population geneticists came forward with the answers. The latest update was released in 2025 in a research led by Professor Livni Skorecki and co-authored by Joseph Livni. Before we discuss the research, let me introduce the authors. Livni Skorecki was born and raised in Toronto, Canada, and received his MD degree from the University of Toronto. He pursued postgraduate clinical and research training in internal medicine, nephrology, and molecular biology at the Brigham and Women's and Massachusetts General Hospital and Harvard. He served as the director of nephrology department at Rambam Haifa, Israel, director of medical and research development, director of the Rappaport Research Institute of the Technion Haifa, and dean of the Bar-Ilan University Azrieli Faculty of Medicine in Tzfat. Carl's research enjoys international interest. In 2000, he appeared on 60 Minutes. Among others, Carl is one of the pioneers of studies related to founding patrilineal and matrilineal lineages in Jewish populations in general, and in Ashkenazi Jews in particular. Joseph Yossi Livni is a graduate of the Technion Haifa. If you guessed that the two co-authors met at the Rappaport Research Institute, you're partially correct. However, it's not because Yossi served on the staff of the institute. When Yossi graduated the Technion in mechanical engineering, Carl was in AHS High School in Toronto, Canada. When Yossi studied as a graduate student at the University of Tel Aviv, Carl carried out his postgraduate medical studies in Massachusetts. When Yossi received his master's degree in engineering at the University of Tel Aviv, Carl was back in Toronto. Yossi moved to Montreal, Canada to work at Bombardier Aerospace in 1986. Carl moved to Haifa, Israel in 1995. No, they never met in Canada. After retiring from aerospace, Yossi realized that his experience in mathematical modeling of complex processes is useful to explore unsolved questions in another field. That field has always interested him: the Jewish community, culture, and tradition. In 2011, Yossi read a research led by Carl. It will be mentioned in this video as Behar et al., 2006. Yossi and his brother Chaim dissected that article with engineering tools. They realized that it can reveal the size of the first generation of Ashkenazi Jews. That number was unknown in 2011. They published their investigation in 2016. This investigation connected Yossi and Carl. Carl knew about the research, met Chaim and Yossi at the Technion, and supported it. Carl and Yossi met at the Technion because they shared an interest: founder populations in general, and Ashkenazi Jews in particular. What is a founder population?
A founder population is a new population established by a very small number of individuals who separated from a larger mother population.
Are all Jewish groups, Ashkenazi, Sephardic, Yemenite, etc., different founder populations?
Good question.
No, not all Jews are founder populations. However, the Ashkenazi Jews are a founder population.
It implies less genetic diversity than a typical population. In other words, at a specific location of the genome, individuals in a founder population display a limited number of variants.
Then what was the larger population from which the Ashkenazi Jews separated? How many were they? Did they live on an island? On top of the mountain? Why were they isolated from the mother population?
The article of 2025 references two earlier works, concluding that all Ashkenazi Jews are descendants of 300 individuals. The two works arrived at this estimate using two different theoretical approaches. The article confirms this estimate using a third approach. It consists of a more experimental methodology. This settles the question of the size of the first generation. As far as where did they settle, there are medieval rabbinical documents testifying that they settled in the Rhine Valley. As mentioned before, the question, where did they come from, needed more research. On this question, population genetics is useful.
Is this a question for geneticists only?
Surprisingly, historians, linguists, geneticists launched several competing theories. One can group them in three main groups. The Khazar theory claims that the Ashkenazi Jews are descendants of the Khazars. The European theory locates the origin of the Ashkenazi Jews in Europe.
The Khazars were Turkic tribe, and some practiced Judaism, weren't they? Why can't the Ashkenazi founders be Khazars?
The Khazar origin theory has proponents among political figures, authors, journalists, linguists, and even geneticists. However, we show here a table of historians, serious population geneticists, linguists who looked for evidence supporting the theory. No such evidence has been found. The theory has been abandoned.
I guess we are left with either Europe or Levant. Were there any Ashkenazi Jews in the Levant?
No, there were no Ashkenazi Jews in Levant. However, there was a Roman province called Judea. Most inhabitants of Judea practiced an ancestral faith, customs, and civic order. The Edict of Augustus granted Jews the right to observe their ancestral customs, protect their sacred offerings, and maintain religious practices without interference from Roman authority. There were sects. However, none of them identified itself as Ashkenazi. The term Ashkenaz is mentioned in the Torah. However, it consistently implies a foreign land or nation. In Jewish sources, the term Ashkenaz is recycled. It is identified as Lothar, reasonably Lotharingia. This is consistent with the presence of Ashkenazi Jews in the Rhine Valley. However, it does not imply where this population came from.
Then the Ashkenazi Jews could be Europeans who converted to the Jewish faith, couldn't they?
Yes, that is the European hypothesis. However, this theory has been partially abandoned because it is inconsistent with genetic test results of male Ashkenazi Jews. Nevertheless, the validity of the theory has been argued as far as female Ashkenazi Jews are concerned. Let us follow the progress of scientific work. It consists of several twists and turns of results and their interpretation.
Does that mean that we have no clue where the Ashkenazi Jews came from to the Rhine Valley?
Actually, there is a study pointing to one location of origin, Lucca, Italy. That traditional Jewish study only focuses on one single founder, Rabbi Kalonymus.
If Rabbi Kalonymus migrated from Lucca, doesn't that indicate a European origin?
Rabbi Kalonymus was a male. The consensus is that males came from the Levant. The map on the right-hand side provides Levantine locations. The map on the left-hand side offers European alternatives. These maps show geographic areas from which the Ashkenazi founders came.
The Levant locations are exclusively compatible with the Levant theory.
The migration of Jews out of Judea started 1,000 years before the Ashkenazi founder event. A 2 or 3-phase migration from the Levant to Italy, France, and Spain is not only possible, but also supported by evidence.
According to Jewish tradition, the Jewish diaspora came from the Levant. How was this scientifically confirmed?
In 2006, an important investigation led by Professor Skorecki, also known as Behar et Altri 2006, showed that 40% of Ashkenazi Jews descend from 4 founder mothers. The lineages of these founders have been called, since then, major lineages. The same investigation found out that the mitochondria of these lineages is also found in non-Ashkenazi Jews. Thus, the common ancestors of these major lineages lived in the shared homeland of all Jews. In addition, this investigation also tried to test the possibility of a common ancestor in Europe. They practically found no mitochondrion identical to the mitochondria of the major Ashkenazi lineages. The Levant hypothesis 1.0 has been launched and demonstrated.
This theory is confirmed by the absence of evidence. Is absence of evidence evidence of absence?
If a hypothesis does not imply existence of evidence, then absence of evidence is not evidence of absence. For example, some archaeologists concluded that there was no exodus from Egypt because repeated excavations in the desert failed to unearth evidence. I wonder what would qualify as evidence of exodus. Unconsumed gefilte fish? Lost pages of the Pentateuch? Buried yarmulke? However, in our case, the hypothesis that claiming European origin of Ashkenazi Jews does imply that sisters or maternal cousins of these hypothetical converts to Judaism have descendants in current Europe, carrying the mitochondrion of the converts. Short of justifying the absence of evidence is evidence of absence.
How did then the Europe hypothesis come into sight? It had to address the absence of the 4 major mitochondrial control region sequences in West Europeans, hadn't it?
In spite of the challenge, a 2013 genetic study concluded that Europe is the origin of female Ashkenazi founders. The study did acknowledge that the mitochondrial sequences of the four major founders are absent among Europeans.
How does the Europe hypothesis explain the absence of the four major mitochondrial sequences in West Europeans?
The hypothesis of European origin of Ashkenazi female founders accepts the previous map as far as the male founders are concerned. It hypothesized the same 2-3-phase migration of males. Once in Europe, these males married converted European females, and according to the hypothesis, in subsequent phases, they migrated to the Rhine Valley.
According to this understanding, a shared origin of males and females makes more sense. The map is consistent with Colin Renfrew's term demic diffusion migration, describing the spread of Neolithic farming. Nevertheless, I prefer the term mass migration. It relates to the biblical image of the Exodus, or the massive post-Columbian European migration to the New World. It distinguishes this type of migration from random migration. Random migration is when the bride joins the bridegroom's community. Gender bias in random migration does make sense. In those patriarchal centuries, one cannot rule out a dominant pattern of the bride adopting the groom's religion. On the other hand, gender bias in mass migration must be rare. Both Native Americans and European migrants arrived to the Americas, male and female of every living thing, as God has commanded Noah. What is the reason for the implausible male-only migration scenario?
The researchers of the British team, 2013, used the phylogenetic analysis of 836 Ashkenazi mitochondrial control region sequences. For each sequence, they assessed its geographic origin. For example, this phylogenetic chart substantiates 6 Ashkenazi mitochondrial sequences having European ancestry.
How many Ashkenazi mitochondrial control region sequences did they analyze? How many turned out to be of European ancestry?
As mentioned earlier, the research of 2013 examined 836 Ashkenazi mitochondrial control region sequences. They found that more than 80% of Ashkenazi mitochondrial control regions are of European origin. This leaves to about 167 non-European matrilineal lineages. Their conclusion was, quote, the maternal lineages mainly trace back to prehistoric Western Europe.
It seems to me that this conclusion contradicts the findings of the research led by Professor Skorecki of 2006 that postulated Levant theory 1.0. If the maternal lineages trace back to the Western Europe, then the four major mitochondrial sequences should be found in current West European population. If a West European bride converts to Judaism, her mitochondrial control region sequence does not vanish in Western European population. There's a mystery here. Either the majority of the maternal lineages don't trace back to Western Europe, or at least some of the four major Ashkenazi mitochondrial signatures should be found in Western Europe. Did they look for them? Did they find any?
Yes, they thoroughly looked for them. They increased their sample size 22,257 Europeans, of which 15,264 were West Europeans. The results yielded no change. Practically, none of the Ashkenazi major mitochondrial sequences have been inherited by the hypothetical non-Jewish relatives of the major lineage founders. Nevertheless, the 2013 research reconciled the riddle.
How?
migration did follow the pattern suggested by the Levant hypothesis.
The males migrated from the Levant to Western Europe. They found West European brides. Hypothetically, none of the brides carried the major mitochondrial CV sequences. That explains why we cannot find their sequence in current West European population.
Then how did these signatures appear as majors in Ashkenazi Jews?
They argued that the predecessors of these signatures do exist in Europe. A predecessor of any control region is its copy minus the last mutation. It was carried by ancestors of the individuals in whom the last mutation took place. It is like the previous model of the control region sequence. Such previous models, control region sequences, are carried by Europeans. The British team postulated European brides that converted to Judaism carried the previous model of the sequence. The new model of each new control region sequence appeared during the 1,000 years period between the beginning of the diaspora and the Ashkenazi founder event. The new model consisted of the addition of a mutation. It was not and could not be transmitted to host population because of strict Ashkenazi endogamy.
Is there supporting evidence to this narrative?
The supporting evidence consists precisely of two contradictory genetic findings. The majority of Ashkenazi matrilineal lineages reach European ancestry. The major Ashkenazi mitochondrial control region sequences are not found in Western European populations. Without this narrative, the European theory falls apart. In 2025, Professor Skorecki and myself published a new investigation, Levant Theory 2.0.
Does the 2025 research led by Professor Skorecki refute the findings that the European matrilineal lineages significantly outnumber Ashkenazi matrilineal lineages of the Levantine ancestry?
25 version of the Levant theory accepts that Ashkenazi Jews belong to a multitude of lineages of European ancestry and a few lineages of Levantine ancestry.
Can the Levant theory reconcile the multitude of European matrilineal lineages with the absence of the major mitochondrial control region sequences?
As explained earlier, the Levantine theory preceded the European theory. It was founded on the finding that none of the major Ashkenazi mitochondrial sequences is found among Europeans. If a European founder converts to Judaism,
many copies of her mitochondrial sequence are conserved in the European population.
This rationale excludes the possibility that most founders were Europeans. The 2025 Levant theory returned to the previous argument. However, it had to address the multitude of European matrilineal lineages detected by the 2013 investigation. The 2025 version of the Levant theory reveals that there is no contradiction between the disproportionate presence of European matrilineal lineages and non-European origin of founders. If the Levantine founder population absorbs European individuals, then absorbed matrilineal lineages are bound to significantly outnumber founder lineages. Thus, the multitude of absorbed European lineages does not contradict the Levantine origin of founders. The initiative of the second Levantine hypothesis was launched by Professor Skorecki in July 2016 at the multidisciplinary workshop that took place in Haifa on July 2016.
Why did it take 9 years to publish it?
The investigation is cross-disciplinary. Cross-disciplinary research is not impossible. However, it takes longer. The investigation required developing and testing a new approach, distinguishing between founder and absorbed mitochondrial sequences. The main reason, however, consists of the fact that Professor Skorecki served during these years as Dean of the Bar-Ilan University Azrieli Faculty of Medicine in Safed. The Dean is busy with oversight of international and national research partnerships, research training, research centers, establishing and monitoring budgets, assisting in securing external funding to enable cooperative research, assisting with improving publication outcomes, and contributing to the strategic management of the faculty. In spite of that, this cross-disciplinary investigation of the refreshed Levantine hypothesis continued under the heavy burden of other priorities.
However, this investigation seems important. The finding of the British team of 2013 spilled into malicious claims of overt and covert anti-Semites. For example, NBC claimed on October 8, 2013, that a research of British team questions the ties of Ashkenazi Jews to the land of Israel.
The right of Jews to their ancestral land is not a scientific question. The British investigation did not question the connection between Jews and their land. Our 2025 work is also a scientific investigation following the scientific method: observation, theory, proof. The NBC or others are not bound by such constraints. The NBC took a publication that fits their inclination. We are scientists and we publish proven science. The NBC and other outlets are invited to read our publication and update their audience. I would like to stop this line of questions and continue to focus on science.
I agree. However, the 2013 article in favor of European origins found very few Levantine mitochondrial sequences and many European sequences.
That is a good question. Let me answer it with another question. If we know that the founders consisted of 150 females, how many mitochondrial control region sequences they displayed?
Let me think about it. I guess 150 unrelated founder mothers had 150 control region sequences.
Very good. The correct answer is C. Now I will ask you a related multiple choice questions. How many founder mitochondrial sequences are in the current generation?
I'll play music while you think about it.
All 150 founder mothers have descendants in the current generation. Consequently, I guess it's C again.
No, it is D. I admit, it is a trick question.
It opens the door to the next section.
Does the theory start with the fact that the first generations consisted of 150 matrilineal lineages?
Yes, it does. We can see here 150 equal dots representing the 150 original matrilineal lineages.
Now let's go to the second generation.
37 mothers had 2 sons and no daughters. 37 lineages became extinct. 37 mothers had 2 daughters. Some lineages grew. Lineage extinction continued. The extinction rate gradually slowed down. The process stopped after 15 generations. As some lineages vanished, others grew. 4 of them grew disproportionately. We call them major lineages. Watch the following illustration. After 15 generations, lineage extinctions of founders tapered off. 4 lineages grew into major lineages. About 50 others survived the extinction process and became minor lineages.
Then what are the hundreds of lineages found by the 2013 research promoting the Europe theory?
Great question. It is time to introduce the term absorbed lineage. Let us consider a Jewish wedding in the past.
Mazal tov.
Ashkenazi endogamy means that both the bride and the bridegroom were Ashkenazi Jews. However, tradition is not a law, and Jewish law does allow brides to convert to Judaism following a prescribed procedure. Some non-Ashkenazi Jewish families also appeared within Ashkenazi communities. Some communities lived side by side, for example, London, Amsterdam, Istanbul, Crete, and Bucharest. These marriages required no conversion or any restriction. However, genetically speaking, the mitochondrial control region sequence of such brides, non-Ashkenazi or converted, added one more lineage to the repertoire created by the founders. We call these additional lineages absorbed lineages.
This means that most of the European lineages detected by the British team of 2013 are absorbed lineages, doesn't it?
Yes, it does. Most of the 668 or 80% Ashkenazi Euro lineages the British team detected must be absorbed lineages. There were only 150 founders and only 54 founder matrilineal lineages survived lineage extinction.
How do we know that Jews in general and Ashkenazi Jews in particular practice endogamy?
Any continuous ethnic group for several generations practices endogamy. In particular, founder populations practice endogamy by definition. As a consequence, in Ashkenazi communities, both the bride and the bridegroom were Jewish.
What does that mean in terms of mitochondrial sequence of the bride?
It means that her mother was also an Ashkenazi Jew, and that went on from generation to generation. The more strictly was endogamy observed, the higher the chance that the bride carries the mitochondrial sequence of a founder.
What happens if the bride is a European who converted to Judaism?
Mazal tov, the bride is a Jew and her descendants are Ashkenazi Jews.
I knew that. However, what about her mitochondrial control region sequence?
Her mitochondrial control region sequence is a distinct sequence unique in the Ashkenazi population. Such a Eurobride adds a new Eurolineage in the phylogenetic display of Ashkenazi lineages. No wonder the British team led by Professor Richards in 2013 found more than 668 lineages that phylogenetically descend from European ancestors. In 44 generations since the founder event, 668 brides of European descent seems like a gross underestimate.
When did this admixture start?
Reasonably, as the population increased and spread, the number of absorbed brides grew. Let me take you to Amsterdam of Rembrandt. Rembrandt lived in Jodenbreestraat in the Jewish quarter. There were 3,000 Jews in Amsterdam. 1,000 of them were Ashkenazi Jews. The rest fled to Amsterdam from Portugal.
How do you know this bride is Ashkenazi?
I guess so, because the artist, a pupil of Rembrandt, titled the painting Esther Bedekt. It is a Yiddish term, meaning Esther veiled, an Ashkenazi wedding custom. If she converted to Judaism to marry an Ashkenazi bridegroom, then she added one new European absorbed lineage to the Ashkenazi repertoire. If she were from Portuguese Jewish community, then Esther added a non-Ashkenazi absorbed Jewish lineage.
How many matrilineal descendants of Esther are currently in the Ashkenazi population?
Good question. In 1650, there were 250,000 Ashkenazi Jews. She added then one in 250,000. Reasonably, her descendants multiplied at the same rate as other Ashkenazi Jews. Consequently, the incidence of her descendants is still 1 in 250,000. Therefore, in the current population of 10 million, Esther has about 40 descendants.
How do we know if Esther was an absorbed Ashkenazi Jew? Can genetics help?
Yes, we can, and this question opens a new subject.
Can you distinguish genetically the mitochondrial control region of the descendants of Esther and of Yentl? How? By phylogenetic analysis, as the British research of 2013? Is Jewish DNA different?
The research of 2025, led by Professor Skorecki, offers an approach of distinguishing between the mitochondria of Esther and Yentl. However, the approach is not phylogenetic analysis. Our approach stems from the previous observation that Esther has about 40 descendants in the current population.
How many descendants has Yentl?
Yentl has also about 40 descendants. However, Yentl was a matrilineal descendant of an Ashkenazi founder. There is a 40% chance that she is a descendant of a major founder. In that case, Yentl's mitochondrion has about 1 million copies in the current population.
How many descendants would Yentl have had if she were a descendant of a minor founder?
If Yentl were a matrilineal descendant of a minor founder, her current matrilineal lineage would be at least 20,000 strong.
I still don't know how this helps to distinguish between the mitochondrion of Esther and of Yentl. I mean, if both are present in a sample of harvested from Ashkenazi Jews, how does Esther mitochondrion reveal that her ancestor converted to Judaism?
Well, Esther's mitochondrion reveals that she is the descendant of an absorbed mother because her control region's sequence appears as a singleton.
Yentl's lineage exceeds
20,000 descendants. As a consequence, her mitochondrion will appear in that sample multiple times. On the other hand, Esther has about 40 descendants. There is a slim chance of one of her descendants to be selected into a sample of 600 Ashkenazi Jews. If such a descendant is selected in spite of all odds, then the mitochondrion of Esther will be found in one single copy. Our approach is to count the singleton mitochondria in the sample. They all represent absorbed lineages like Esther's. Thus, one can establish the proportion of absorbed lineages in the population. In a sample of 575 Ashkenazi Jews, the expected number of the mitochondrion of descendants of founders is about 8. The expected number of copies of the mitochondrion of descendants of absorbed foremothers is about 0 to 1.
How does this Levant theory 2.0 reconcile the multitude of European lineages with the absence of major Ashkenazi lineages in Western European population?
There is no need for reconciliation.
In a founder population with less than 100% endogamy, the natural outcome is a disproportionate number of absorbed lineages. The absence of mitochondrial control region sequences in Western European does not need any explanation. This absence is the observation that gave rise to the Levant theory 1.0 of 2006.
Does the 2025 article mention the narrative of the European theory 2013 reconciling this same contradiction?
It does. It estimates the plausibility of the scenario of 4 major lineage mutations in 40 generations. There is very little likelihood of that happening.
Does it mean that it couldn't happen?
If that scenario were the only explanation of the coexistence of many European lineages and absence of West European major mitochondrial control region sequences, then even a minimal probability could not be a reason to dismiss it. In view of the fact that there is no contradiction between the two observations, one can say with confidence that that scenario is fictional.
What does the 2025 Levant 2.0 article prove?
Mitochondrial sequences of founders appear in multiple copies. Mitochondrial sequences of absorbed foremothers appear as singletons. The founders descend from Levantine ancestors because practically no major founder mitochondrion has been found in West European populations. That indicates non-European ancestry of Ashkenazi Jews. An alternative explanation for this fact, suggested by Costa et al. 2013, is extremely implausible. The previous determination of the Levantine origin of the founders of Behar et al. 2006 is valid.